இப்ப கரெக்டா இருக்கு.
இது இருக்குது
சரி, அந்த காலர்ல தான் பேசணுமா? காலர் மைக்ல. சரி, செஷன் ஸ்டார்ட் பண்ணிடலாமா? மோகன் ஸ்டார்ட் பண்ணிடலாமா?
ஓகே, சார் லைன்ல இருக்காரு, அவர்கிட்ட கேட்ட ஒன்று சரிங்க, ஸ்டார்ட் பண்ணிடலாம் விடுங்க.
சார் குட் மார்னிங் சார். அர்ஜுன் சார். மசூர் கான். அர்ஜுன் மசூர் கான். டாக்டர் அர்ஜுன் விஜய் மசூர் கான்.
ஹலோ
சார்.
ஹலோ.
வாய்ஸ் அவரால் ஹேர் பண்ண முடியலைங்களா?
என்னோட வாய்ஸ் கேட்கலீங்களாமா? சார் என்ன ஒரு டைம் உங்களுக்கு கேக்குதுங்களா?
எனக்கு கேக்குது
சார். ஒரு டைம் மீட்டிங் லீவ் பண்ணிட்டு திருப்பி ரீஜாயின் பண்ண சொல்றீங்களா?
டெக்ஸ்ட் பண்ணிட்டு இருக்கேன் அவர்கிட்ட, அப்படியே இருங்க.
மோகனோட வாய்ஸ் கேக்குது. மோகன் யூஎஸ்ல தான் இருக்கு. மோகன் உங்க வாய்ஸ் சார் கேக்குதா? அதுவும் கேக்குறீங்களா?
இருங்க, நான் அவர்கிட்ட இன்னொரு போன்ல இருந்து அவர் கால் பண்ணி பேசுறேன்.
சரிங்க. ஆடியோ ஆஃப் பண்ணி வச்சிருந்தாருன்னா மேபி மியூட் பண்ணிருக்காரா? இல்ல மியூட் பண்ணிருந்தா நம்மளோட வாய்ஸ் தானே கேக்கலப்பா.
அவர் லைன்ல இருக்கிற மாதிரியே வரல. ஹெட்லிங் இருக்குதுங்களா?
4 பேரும் காமிக்குது ஆக்சுவலா, ஆனா பார்ட்டிசிபென்ட்ஸ்ல அவர் வரல.
Arjun, this is Mohan. Yeah, are you in line?
I am. I have logged into because the host.
Okay. Yeah, actually, you can hear their voice. Okay, I say to them, they need to allow the host. Okay, I can talk, I can hear their voice. Okay, one second. Actually, நீங்க வந்து அந்த ஹோஸ்ட் ஆன் பண்ணி விடுங்க, அவங்க அலோ பண்ணி விடுங்க.
ஃபுல்லுமே அவருக்கு ஆக்சஸ் இருக்குங்க மோகன்.
Okay, I think you have access. Okay,
நம்ம Zoomல தான் நமக்கு அந்த மாதிரி பெர்மிஷன்ஸ்லாம் கொடுக்கணும். Google Meetல when we are letting you inside you can present actually.
Can disconnect and reconnect. Yeah. Yeah. I'm sending to you. Yeah, that link.
இது அங்க இருந்து எடுத்துட்டு வந்துட்டேன். இந்த மைக் தான் கேக்குமா?
Yeah. I'm sending to you email also.
அதுதான் இப்ப லீவ் பண்ணிருக்காங்கப்பா, வருவாங்க.
Yeah,
Okay. 11:30 நினைக்கிறேன்.
அவருக்கு வந்து ask to join கேக்கு தான் பிரசன்ட் பண்ணலாம். எந்த இதை கிளிக் பண்ணனும் சொல்லுங்க.
ask to join கொடுக்க சொல்லுங்க.
ask to join. Yes. Join பண்ணிட்டாரு.
Okay, there we go. Okay, now I can hear you.
Okay.
So, good morning, sir. Are you able to hear us now?
Yeah, I can hear you. Can you hear me?
Yes, sir. We are able to see you. Yes. And when you're going to do the presentation, kindly go to the present option, and from there you will be able to present the PPT, sir.
Oh, it says present now.
Yes, sir.
Present now, yeah. Okay. Yeah, let me know when.
So we'll start with the formal introduction, and shall we move to the presentation part?
Me?
Yes, sir. So we'll start with the formal introduction.
Okay.
Thank you, sir.
Sure.
Good morning, dear ladies and gentlemen. We extend a warm and a pleasant welcome to everyone on this cheerful morning. We are pleased to welcome the staff, the students from various colleges, We had a delightful and enlightening experience yesterday listening to the talks of the experts. Likewise, let us enrich your knowledge from today's technical session too. We extend a very warm welcome to Dr. Arjun Masarkar sir, who will be taking on the technical session 5. And to introduce Dr. Arjun Masarkar, we are having our head of the department, Dr. G. Nidhirajan sir. I'm handing over the mic to Dr. Nidhirajan sir for the introduction and for chairing the session too.
Good morning, one and all present here. I am Nidhirajan, head of the department, PG and Research Center in Biotechnology, MGR College, Hosur, India. I am honored to introduce Arjun Vijay Masrukar, MD, PhD, in the International Conference on the Genetic Disorders in Human. Professor Arjun Vijay Masrukar did his undergraduate degree in electrical engineering and MD in medicine and PhD in neuroscience. And he did a postdoctoral fellow in Columbia University, and he has a lot of certification, license, and he has appointed many various positions like instructor and co-director, director, investigator, and principal investigator, and he has got many funds from various funding agency, and he got many awards and honors, and he occupied many positions like director, etc. He has published more than 32 research articles in peer-reviewed journal, more than 10 books and reviews, more than eight papers in under review process, and manuscript in preparation is about 10 articles. He has submitted 61 abstracts in various conferences and symposium. With this brief intro, I hand over the presentation to Professor Arjun Vijay Masrukar. Thank you, sir.
Well, thank you very much for that very nice introduction. Can everyone hear me?
Yes, yes.
Yeah, okay, great. And I will go ahead and share my screen. Oh, I see. Just do this here. Okay, can everyone see the PowerPoint? Yep. Great.
Yeah, I can see.
Okay, so I'm really happy to talk to you all today about my talk titled "Towards a Molecular and Cellular Epidemiology of Early Alzheimer's Disease." And I have no disclosures. And first I wanted to acknowledge the hard work of my team. I have both a basic science and a clinical laboratory at NYU, and I have a lot of colleagues that I collaborate with as well. And I'm going to really be highlighting the work of Isabel Reyes and Chengju Tian in my lab. And of course, Mohan Kumar Tangavel has also joined my lab last year, and he's on the call as well. We're very happy to have him at NYU in my lab. So the agenda of my talk is broken down as follows. First, I want to give you all a brief primer on what is Alzheimer's disease so that we're all on the same page. Then I'm going to talk about Alzheimer's disease susceptibility at a macro scale, a large scale, really focusing on the entorhinal cortex and area CA1 of hippocampus as vulnerable regions. Then I'll talk about really honing in on the area CA1 of hippocampus discussing the functional heterogeneity of subpopulation of neurons in that area. I'll then talk about the differential effect of Alzheimer's disease on those subpopulations in experiments that we've done in transgenic models of AD. And lastly, I'll end with some data on the differential and molecular neuropathology of CA1 subpopulations in human Alzheimer's disease. And for those who don't know the pictures that I'm showing to the right, in the upper left picture is Dr. Alzheimer, for whom the disease is named. He was the first person to really describe the pathology of Alzheimer's disease, how it's defined today, and the first patient in whom he described it in is depicted on the right. And below is a cartoon of the hallmark pathologies of Alzheimer's disease, the sort of cottony looking amyloid plaque below, and the tau-based tangle, sort of the dark triangle appearing neuron above that. So first, a brief primer on Alzheimer's disease. We talk about the word Alzheimer's, we talk about the word dementia. What does it mean? I think we have to really contextualize for the rest of the talk. So dementia is a loss of cognition or behavioral abilities that were previously acquired to the degree that one loses their independence. They're not able to perform normally in their daily life, social functioning, or at their occupation. The pathway to dementia is a series of stages going from normal to end stage. And it's a progression. So you have normal at the top, preclinical, meaning there are symptoms or biological signs. So there are no symptoms, but there's some biological signs. We then have a very important, yeah. Okay, sure. Presentation mode. The presentation mode. Yeah. Yeah, where do I get the presentation mode? Yep. A couple example, yeah. Yeah, that's what I clicked. Do you see that?
Yeah, able to see mobile.
Ah, okay. I mean, right now, do you see kind of like the minimized screen? Sure. Yeah, I can do that. Sorry about that. So I'll do the stop presenting. And then what I'll do is, I guess I'll open up my presentation first. Maybe, then I'll go to my slideshow. Let's see. Nope. Show presenter view. Maybe that's what I need. Maybe that's what it is. So what we'll do is, I think I got it. Window. We'll do PowerPoint. Allow. And then we'll go here. And then we'll do this one. Is that better?
Can you hear?
Yeah.
Yeah, is it fine? Yeah,
Uh,
I can see in my mobile his presentation slide also I can see. Okay.
huh, it's actually in the presentation mode. Oh, okay. That's okay. At least I'm not the only one. Select slideshow tab, and then okay. Maybe this is what I should do. How about now? Now? Uh-huh. Oh, you know what? I accidentally stopped the sharing. But I think I know. I think I'm supposed to hit the presenter view in the slideshow menu of the PowerPoint software. Um, then I can go. I can share either a window or my screen, but I'll pick a window. Okay. How about now? And then how about this? Does this work? F4? No. Ah, Shift F5. Okay, I did that. Oh, how about this? How about this? You know what? There's another way that I can do this too. If I, so I can either present, I can actually share my entire screen. So if I share my entire screen, I think that should work. Well, let's try that. Share my entire screen. Let me just make sure I, yeah, entire screen. So you'll see this, but then I will then take over the whole screen with the PowerPoint. How about that? Okay, all right, we got it. Okay, on the fly. Okay. So where were we? Yeah, so dementia really is a spectrum going from normal to end stage. And so there's a critical stage in between called mild cognitive impairment, where there is a little bit of objective cognitive deficits, but the person is relatively intact with their activities of daily living. And then so on and so forth. You get the dementia stages after that, until there's very little functioning in day-to-day life and at home. Now, the causes of dementia are quite broad, and Alzheimer's disease is the most common, which is why you hear about it the most. It's about 60 to 70% of all of dementia cases. What is Alzheimer's disease biologically? It's defined by its neuropathology. And in essence, there's two main pathologic proteins, amyloid plaque and tau-based neurofibrillary tangles. The amyloid plaque is outside of the brain cell, and the tau-based tangles are inside of the neurons. Now, the problem is that Alzheimer's disease prevalence is growing at a very rapid rate, as you can see from the chart here. And so this is a huge problem. in terms of burden on society, financially, and also a lot of burden on the caregivers. And so we really need new treatments to slow Alzheimer's disease progression. So what do we know about Alzheimer's disease? Well, this graph sort of encapsulates what we know about human Alzheimer's disease. We know that there's a series of processes which we think begins with the rise of amyloid. And this rise in amyloid may even begin in our 50s, as you can see by the leftmost red curve. After that, you get elevation in tau, then you get destruction of brain structure, followed by memory loss, and followed by loss of clinical function. Um, Now, in the beginning, a lot of the trials had focused on dementia stage and also the MCI stage, but a lot of those trials have failed. And so now the scientific focus has really been shifted towards the impairment, preclinical, and cognitively normal um, stages of the disease. And so we really need to understand the disease at these early stages. So in that context, I will talk about susceptibility to Alzheimer's disease in these early stages. So MRI technology had allowed us during the 2000s to really establish that the medial temporal lobe is involved early in the disease. And so as indicated here by this coronal MRI section, the hippocampi and the parahippocampal gyrus, which are indicated by the arrows, become smaller in Alzheimer's disease, especially early on. And the smaller those volumes are, the more likely the person is to progress. So this seems to be a critical node of early Alzheimer's disease. Now, what does it correspond to neuropathologically? So there's this staging of Alzheimer's disease called the Braak stage, and it goes from 1 to 6. Now, loosely, normal people are going to be Braak stage 0, where there's very minimal or no pathology in the brain at all. People who are mild
Students, can
cognitive
you please
impairment,
settle
who have
down?
pre-dementia,
The talk
basically,
is going
are
on.
going
Kindly
to
please
be
settle
Braak
down.
stage 1, 2, or 3.
Please
And
don't
that's
disturb
where
others.
the tau
Settle down.
pathology is really restricted to the entorhinal cortex and the hippocampus. Once you start to get to Braak stage 5 and 6, these are largely going to be people with dementia stage of Alzheimer's disease. So when we're talking about early stage Alzheimer's, we're really focused on stage 1 through 3, where we are really referring to entorhinal cortex involvement and hippocampal involvement. But even within hippocampus, there's a susceptibility, and that's the CA1 area. So to the left is depicted the hippocampal formation in human, and as you can see that there are 5 domains of the hippocampus: subiculum, CA1, CA2, CA3, and CA4. CA1 is the main output area of the hippocampus, and it seems to be the part of the hippocampus that's affected the most early in the disease and most robustly by the pathology. And this is evidenced by the panels that I've shown here. And so you can kind of look back at that Braak staging and more precisely indicate that this sort of mild cognitive impairment stage probably corresponds to the transition of pathology from entorhinal cortex to CA1. So that's a critical gateway in this disease. And then it moves from CA1 and beyond. So we really need to get a handle of what's going on within area CA1. Now, when you look at a microscopic level at area CA1, you will see that not all of the neurons are exhibiting pathology. So here's an example of a brain from a 72-year-old man with Alzheimer's disease, focusing in on the neurons within hippocampal CA1. And what you can see is that there's a tangle in one neuron, but the neuron right next to it does not. And so why does this happen? Why does one neuron have the pathology? Why is it affected? And why is the other neuron not? And so what are the rules that would really dictate why this happens? And so I kind of look at it through an epidemiology lens. You know, when you look at epidemiology of a disease at the level of populations, you talk about risk factors. And those risk factors are related to who or what you are, right? Where you are, where you live, and what you're exposed to. And so you can take these concepts and you can really apply those to cells, to neurons. So what do your input connections as a neuron, how do those relate to your risk factor of becoming involved in Alzheimer's disease? Where does your anatomical position within CA1 relate to your dysfunction due to Alzheimer's disease or the pathology that may arise? And what does it have to do with who you are, your intrinsic physiologic properties or your genetic molecular properties? And so this is the lens through which I conduct my research in Alzheimer's disease. And so owing to this framework, I wanted to speak to the functional heterogeneity of CA1 subpopulations. And so just to preface that, I wanted to review briefly the canonical neural circuit of area CA1. So what I've depicted here is a pyramidal neuron of CA1, the main neurons, and it receives input from various areas in the hippocampus and outside. So the entorhinal cortex gives inputs to the distal dendrite of the neuron, and in that same location, the thalamic inputs from nucleus reunions of thalamus come in as well. Area CA3 of hippocampus relays dentate gyrus input at the proximal dendrite, and the basal dendrite receives information from area CA2. Now, all of this information, other than the thalamus, is routed through entorhinal cortex. So entorhinal cortex provides inputs both directly and indirectly via dentate gyrus in CA3 and CA2 to area CA1. So you have direct pathway and indirect pathway. There are multiple anatomical axes to the area CA1 of hippocampus. So one axis is called transverse, that's going from subiculum to CA2. One axis is called longitudinal, which is in the mouse dorsal to ventral. In human, it's called posterior to anterior. And then you have the radial axis, which is the depth axis. How deep in the brain is the neuron? And so there have been a lot of studies, including from my lab, in how does the functioning and the properties and the genetics of these neurons vary across these anatomical axes. And what we found is that there's more spatial information in the proximal side of CA1 when we're looking at the transverse axis towards CA2. And so non-spatial information, sort of olfactory responses, for example, are found more in distal CA1, which is towards subiculum. There's a hypothesis that the transverse axis function, the sort of dichotomy between spatial and non-spatial function, derives from the entorhinal cortex input that comes to those regions. And so it's hypothesized that proximal CA1 is driven by the spatial part of entorhinal cortex, or medial EC, and whereas the distal part of CA1 is driven by the lateral EC, which is specialized for non-spatial inputs. And so what we did is we used mouse in vitro slice physiology and optogenetics to test that hypothesis. And what we did was we expressed the light-sensitive ion channel rhodopsin 2 in axons of either medial or lateral entorhinal cortex. And then once we made our in vitro prep, we recorded intracellularly from neurons in proximal or distal CA1 and used light to stimulate the MEC or LEC inputs and recorded the responses. And what we found was quite striking. Indeed, the medial entorhinal cortex gives very strong excitation to proximal CA1, but very minimally to distal CA1. Whereas lateral entorhinal cortex is the opposite. It gave a very powerful excitatory input to distal CA1, but a very small input to proximal CA1. So this basically proved the hypothesis that both anatomical and functional studies had put forward. There's also the radial axis I mentioned. Now the radial axis is also quite interesting because there are molecular studies that show that these layers are distinct, as you can see from the listing of genes that are differentially expressed in these two layers. But there's also evidence that these two layers are functionally distinct. So similarly, spatial information seems to be more heavily expressed not only in proximal CA1, but in the deep layer, whereas non-spatial responses, olfactory, for example, features of objects, are found more in the superficial layer. And so again, similar to the transverse axis, we hypothesize that radial axis function may derive from the character of its efferent input. And so using a similar optogenetic strategy, we now recorded from deep and superficial cells with channel rhodopsin expressed either in medial or lateral entorhinal cortex. And what we found to the right is that medial entorhinal cortex preferred to excite deep neurons, preferentially in proximal CA1, of course, and lateral entorhinal cortex preferentially excited superficial layer neurons in distal CA1. So not only did the inputs determine the proximal distal functional difference, but they also dictated the deep superficial difference. Us and other groups have also found that the deep layer receives more synaptic inhibition. So as some of you may know, the neurons can either be excitatory or inhibitory in the brain. And so interneurons, specifically parvalbumin interneurons, seem to favor deep neurons, the deep pyramidal neurons. There are other interneurons that are more associated with superficial cells, superficial neurons. In addition, the superficial neurons appear to inhibit deep neurons via those same PV interneurons. And thus, what we and others find is that the deep neurons actually display lower excitation to inhibition ratios. When it comes to intrinsic properties, intrinsic excitability, the deep neurons actually show an enhanced intrinsic excitability. So their ability to fire action potentials, which are the main currency in the brain, are much higher than superficial neurons, both in proximal and distal CA1. And this seemed to be related to basic properties of their intrinsic firing. And our data sort of suggests that there may be certain ion channels that may be differentially expressed to give rise to that increased excitability. We've also found, similar to other groups, that deep neurons show more activity in vivo. So it appears that although that they may have more inhibition synaptically, the enhanced intrinsic firing wins out. And so we've used miniscope imaging, which essentially means these are implantable microendoscopes that can pick up fluorescent activity in the brain. And so we can express through an AAV vector the fluorescent calcium sensor GCAMP within CA1, and then use the miniscope cameras to pick up the fluorescence, which is representative of calcium activity in neurons of interest during a behavior. And so what we found is that when a mouse is moving along a track, it's the deep layer neurons that show more activity and more firing, and more firing specifically in accordance with spatial exploration. And so this again shows that it's actually the intrinsic excitability of the deep neurons that win out over the differences in synaptic excitation. So now that we had been armed with this physiologic information and connectivity information, we then pursued work in Alzheimer's disease models. So we used the triple transgenic Alzheimer's disease model, which expresses both amyloid and tau. And so we performed slice physiology recordings at an age where there should be both pathologies. And what we found was that in terms of intrinsic excitability, in these triple transgenic mice, firing rates were reduced in the superficial neurons, and they were increased in deep neurons. And we find that there's this slow after hyperpolarization, so the suppressive potential that is typically associated with aging was preferentially enhanced in the superficial layer as opposed to the deep. And so what this shows is that Alzheimer's disease pathology, regardless of where it is, appears to impact the superficial layer more than the deep layer in terms of inhibiting their intrinsic ability to fire. And in an almost a compensatory way, the deep neurons are hyperactivated. When it comes to inhibition, we also see changes that promote excitability of the deep neurons. So as I mentioned, deep neurons are preferentially receiving inhibition. These same interneurons are actually affected by amyloid in a way that would suppress their activity. So if there's less inhibition, there's more net excitation of these deep neurons. And this would hypothesize that the excitability of deep neurons is even more enhanced. When it comes to excitation, excuse me, we focused primarily on the entorhinal cortex excitatory inputs. And what we found in our patch clamp recordings was that there was a selective suppression of entorhinal cortical excitation onto superficial cells compared to the deep neurons. The deep neurons were essentially unchanged, as shown in the upper right panel. And we recapitulated this same data using not only electrical stimulation, but also our optogenic paradigm, where we selectively stimulated lateral and medial entorhinal cortex inputs in both distal and proximal CA1. So it didn't matter where along the hippocampus, the CA1 transverse axis the neuron was, the superficial layers seemed to be more vulnerable to loss of input, and the deep neurons were more protected. And the mechanism we find appears to be both loss of spines, so it's a postsynaptic effect that's spine dependent, but there's also a postsynaptic spine independent mechanism as well. And we found this to be true both in older triple transgenic mice, younger triple transgenic mice where there's only amyloid, as well as the 5XFAD model, which has only amyloid. And so this appears to be an effect that's really stimulated by amyloid alone. When it came to the indirect inputs of entorhinal cortex, the CA3 inputs, we found a similar selective vulnerability that also appears to be amyloid driven, but this effect was primarily spine loss independent. So in sum, these data show that intrinsic vulnerability is a hallmark of superficial neurons, and that deep neurons are resilient to the physiologic and synaptic destructive impact of Alzheimer's disease. I was also interested in going back to some of the genetic differences between these two layers to see if there are any clues underlying this differential susceptibility. And lo and behold, two of the genes have been well studied in relation to Alzheimer's disease effects. For example, the serotonin receptor gene, HT1RA, has actually been found to promote amyloid-induced tau phosphorylation. Furthermore, there seems to be a differential expression of the CGN kinase in the superficial layer. And this kinase has been shown to facilitate tau phosphorylation and amyloid-induced spine loss. And so there actually is a molecular genetic mechanism, perhaps, for the differential susceptibility that we're seeing. Now, that's all said and well in the mouse models, but what about human? We always have to recapitulate or affirm our findings in human AD. So it's quite interesting. So I had shown before that the lateral entorhinal cortex appears to primarily innervate or excite distal CA1, and medial entorhinal cortex primarily innervates, excites proximal CA1. And lo and behold, studies that have actually paid attention to this anatomy have actually found that lateral entorhinal cortex is affected before medial entorhinal cortex in terms of AD pathology. And following suit, distal CA1 is affected before proximal CA1. And so this connectivity appears to mirror disease. And this explains some functional changes that we see in patients, we usually see these non-spatial, for example, olfactory deficits before classical declarative memory deficits. So LEC distal CA1 circuit dysfunction before MEC proximal CA1 circuit dysfunction. This type of relationship has also stimulated thoughts about synaptic spread of pathology, since synaptic connectivity seems to be so important. However, I wasn't completely accepting of this hypothesis because I think that there's a lot of evidence for intrinsic vulnerability. So one problem is the lateral entorhinal cortex also targets dentate gyrus, but dentate gyrus, as well as CA2 and CA3. But these three regions are not affected until later. So how could it be that connectivity alone dictates vulnerability? It has to be at least suggesting a strong involvement or co-involvement of intrinsic factors. So what we did was we utilized the NYU Brain Bank and looked at Alzheimer's cases and quantified the degree of amyloid pathology and tau pathology in these two layers, as well as in proximal and distal CA1. And what we found was that, yes, and these were advanced cases. These are Braak stage 5, 6 cases. And what we found is that, yes, there's a slight elevation of pathology in distal CA1 versus proximal, so LEC versus MEC innervation. But really, the gradient wasn't that high. Secondly, when it came to deep superficial, there was a significant increase in pathology in the superficial layer of both amyloid and tau pathology, whether you were in proximal CA1 or distal CA1. And so this actually suggests that connectivity, meaning who you talk to, who you receive input from, is not the primary mechanism, but that intrinsic vulnerability is more important. We then wanted molecular evidence of this hypothesis. And so we turned to a proteomic method. So we are able to use a technique where we can take fixed human AD brains, and we use Braak stage 6, and we can use a specialized microscope called a laser capture microdissection microscope. And we can actually cut out the superficial layer and the deep layer. And then we can use label-free quantitative liquid chromatography mass spec to look at the entire protein ensemble expressed in those layers, and then compare the expression of proteins in one population versus the other. So this method was quite fruitful. We were able to quantify over 3,000 proteins spanning 6 orders of magnitude in abundance. And we are actually able to capture a lot of excitability-related proteins, over 15%. Remarkably, even at an advanced stage of Alzheimer's disease, we found that deep superficial layer differences were still evident. And in fact, one marker, alpha internexin, which was increased in the deep layer, has been shown to be a marker of late as opposed to early vulnerability. And so this actually supported our hypothesis that superficial neurons are affected, may degenerate, develop pathology earlier in the disease, and the deep layers are more resilient and develop pathology, gain dysfunction later in the disease. And so just to conclude my talk, my conclusions are as followed. 1, hippocampal CA1 is affected early in AD, but not all neurons are equally affected. 2, CA1 neurons are intrinsically, functionally, and synaptically heterogeneous across the distal proximal axis and the deep superficial axis. Distal CA1 and superficial neurons therein are driven by lateral internal cortex, whereas proximal CA1 deep neurons are driven by medial internal cortex, giving rise to non-spatial versus spatial functional specialization. Within CA1, Alzheimer's disease leads to hypoactivity of the superficial layer and hyperactivity of the deep layer, regardless of transverse axis position. And then within CA1, AD pathology and related molecular signatures of disease indicate an intrinsic vulnerability of the superficial layer. And these data in total suggest that intrinsic more than extrinsic factors play a role in superficial neuron vulnerability, keying in, owing to the theme of this conference, on molecular genetic factors. And so I'll stop there, and I'm happy to take any questions. Sure. So this will be in the chat. Anybody have questions? Good question. So the question is, as people age, they get memory loss. At what point of time should they visit a doctor for Alzheimer's disease or other diagnoses? That's an excellent question. So I see patients in my practice at all levels. I see a lot of people who are in their 50s, 60s that are perfectly fine, but they're a little bit worried because their mother or father had Alzheimer's and they're worried. So I think it's fine to see a doctor at any point. Now, when do we become really worried? Well, if you're starting to make mistakes every day, that especially noticeable to colleagues or family, then I think for sure it's time to see a doctor. But there's a lot that we can do and modify, even if there aren't so many noticeable symptoms. And I often view myself not necessarily as just diagnosing dementia or Alzheimer's disease, but also helping people optimize their brain health, even in the absence of these diagnoses. Mm-hmm. Great question. So, sir, as you said, only few neurons are tangled, others are normal. Is there a way to untangle the troubled ones? So I'm going to answer this two ways. One is we don't know if the other ones are really normal. Because as you saw, I think it's the superficial ones that become tangled. But the deep neurons are compensating in a way, they are changing. And I don't know if that's a good thing. So one could ask, apart from treating the tangle, should we also treat the other neurons that are not tangled, but maybe not working properly? Now, when it comes to untangling, there have been some strategies out there that have been tried in trials that have been successful. For example, using an antibody or using an agent that can prevent the formation of the tangle itself, for formation of the three-dimensional tangle. They haven't really been successful. I think tau is even harder than amyloid to treat because it's intracellular. I would say that maybe an avenue is to intervene upon the phosphorylation of the tau, because we know that the dominoes start to fall once the tau is phosphorylated in an inappropriate way. So I think intervening upon that process is one way before it becomes a tangle. Huh, that's okay. So is the number of tangled neurons related to the severity of the disease? Excellent question. And just to preface this, right, so there's two pathologies, amyloid and the tau pathology, the plaques and the tangles. And they both have stages. One is called the Thal stage, one is called the Brock stage, was what I showed. The Thal stage, where and how many, doesn't seem to indicate the symptom of the patient, but the where and how many of the tangles does. So to answer this question, yes, it's actually the tau tangles that determine the severity, meaning where they are, meaning the location would tell you which symptom, and how many there are will tell you how bad the symptom is. Thank you so much. Well, thank you for the invitation. This is a pleasure, and sorry for the technical issue in the beginning. Okay, take care, everybody. Bye bye. Good night. Yes. Bye bye.
இல்ல பரவாயில்லைங்க பரவாயில்லைங்க. சரி ஓகே குட் நைட். ஓகே டேக் கேர். சரிங்க பாய். மொத்த அவர் என்ன கரெக்டாக மாட்டேங்குது.
Kitty, are you talking to me? Are you talking to me, Kitty? Kitty, can you hear me? Yep. I am. Okay. Can you hear me, Kitty? I can hear you really well. I can't see you though. Oh, that's good. That's good. Can you hear me, Kitty? Okay. So I have to speak up. Okay. No, no problem. No problem. Is this fine or should I speak a bit louder? That's fine. Don't worry, Kirti, because I have a naturally loud voice, so we will be okay. It, uh-huh. Absolutely, no problem. Kirti, I'm going to have to come out of this and I'm going to have to go into the presentation link, isn't it? Mhm. Yeah. Oh, yes, I do. I do. Oh, yeah, and share. Okay. Uh-huh. Uh-huh. Oh, yes. Yes. Uh-huh. Yep. Okay. The entire screen. Yep. No problem. No problem. Absolutely. No problem. Kirti, just in case if there is a technology problem, you have my presentation. So just all you have to do is read the presentation. I will come back and join you if there is a tech blip. Okay? Okay. So am I going to come out of this and then rejoin you after 11? I'll do that. I'll do that, Kirti. That's much better. Why are you giggling? Go on. Tell me, Kirti. Amuse me first thing in the morning. Go on. Right. Okay. That's fine. Thank you. Yeah. So Kirti, I'm just going to switch off the video and the audio, and yeah, and then when it's time, anyway, give me a shout. I am here, so I'm not going out of the room, so we can just start. All right? Perfect. Yeah, lovely. I will see you in a bit.
Hello. Hello.
కాదు బయట 10:50 answer. Dear students, we shall now break for tea. Kindly please have your coffee or tea which is served outside, and we'll be again rejoining in another 10:50. All of you should be back here. Okay? Students, whoever you're going to present your oral presentation or poster presentation, after the tea break, kindly assemble in seminar hall 2 for oral and poster presentations. Thank you. Please, you can now break for the tea.